Okay, welcome. All right, this is my first recording for you guys. This is going to be our chapter 11 in our Bertino textbook. And this is forensic entomology. So if you remember from the beginning of the year, I'm just going to take my pen out here, that entomology is basically going to be our study of bugs. Okay, so bugs. And we had a little experience with that in the beginning of the year with our decomposition of our squirrel, where we looked at the stages of the blowfly. Specifically, we were examining the maggots and how that worked on decomposing our squirrel. Okay, so on our next slide, if I can manage to go through the next slide. All right, so then how do we interpret evidence, right? So evidence, environmental conditions, and especially to investigate the post-mortem interval, which is PMI, which we will talk about a little bit later, which you might have heard in a TV show or something before. Okay, the vocabulary associated with this unit, and you will have a Google form quiz on this vocabulary when we get a little bit closer to the end of the unit. All right, let me just grab my eraser because that's a terrible kindergarten handwriting. Okay, and I'm going to switch back to pen. All righty, switch back to pen. All right, introduction. All right, so the insects have three basic body segments that you need to be aware of, right? The head, the thorax, and the abdomen. I think it's pretty self-explanatory, right? Head is obviously going to be here, right? And then you really just got to be able to figure out thorax and abdomen. So abdomen is like the larger part of your body, right? So the abdomen is the larger part of this body. And then sometimes we include the wings. All right, so forensic entomology, right, is basically the study of insects, bugs, right, to provide evidence in civil and criminal investigation. And obviously, we have here a picture of maggots, and they cluster on only decomposing flesh. Okay, they don't do anything to living fresh tissue. And remember, they're clean, and they do an efficient job of breaking down the body into its component parts and digesting the body parts. So it's really actually very fascinating and not really as disgusting as you want to think it to be. All right, so we can use the insect evidence to locate a body, right? So basically what insects are on the body, we can determine the location. We can be able to see swarms of insects and be able to determine if a body is present. We can be able to determine if a body was moved based on the types of insects and what the insects are ingesting. And we can identify a geographic range, right? So if a body was transported over a larger distance, right? Again, we can use the different colonies of insects that will colonize the body to determine that kind of a geography, right? You can link a suspect to a crime, right? Because they are going to be there's a lot of evidence that you can then extract from the maggots especially. And you can also have trace origins of drugs. So a little bit of a grossness here. So you can take a maggot colony from a decomposing body of, whether or not that be, human body. And you can basically kind of puree that, and then you can be able to extract the drugs, and you can see like what drugs are present. The forensic entomologist answers some really basic questions. So where on the body is there injury, right? So where was the body moved after death, right? That's important, right? So then that's the difference between the primary crime scene and the secondary crime scene. And was the victim restrained while alive? Was the body covered, buried, or submerged in water? You're going to have a much different colonization of critters. All right, so as way far back as 1247, right, we have associated insects and murder and discovery. 1855, insect evidence was used to solve the death of a murdered infant hidden in the wall of a house. In the early part of the 20th century, research done on the effect of the screwworm flies had on living cattle and sheep laid the groundwork for future entomologists. So entomology is a very large part of forensic science. A decomposing body follows a regular pattern in predictable changing habitats. So we have the fresh, bloated decay, active decay, and then dry. So we have a fresh decomposing body. Really, no decomposition is really taken place, right? The heart has stopped, the body starts to cool, right? So then that's like the first thing. Then we have our bloated stage, right? So bloated just means like full, right? If you're bloated, right, you're basically full of gas, right? So this is where bacterial creatures start building up and producing gas. And this is where you have some, more yucky stuff, right? Then you've got your decay period, right? This is where you're going to get your insects in here. And then you've got your active decay, right? So then this is where the insects are really going to town. And then this is the part where there's really just no insect activity left. There's really no flesh left, mostly just skin and bone at that point. All right, so then nature does a really good job of recycling the nutrients, right, in the decomposition process. So different habitats support predictable sequence of insect succession on a dead body, right? That's going to depend on the location, what type of flies, etc. All right, so the odors of decaying flesh attract multiple bottle flies to lay their eggs, right? So the flies come in, they lay their eggs, right? And then you have kind of start this whole process of the bug life cycle. The two basic odors of decaying flesh are cadaverine and putrescine, right? Those are the chemical names of those two. And I think those are very aptly named. All right, so we have the blowflies, right? We call those blowflies or bottle flies. When the putrescine and cadaverine, right? Those are those two, the chemical names of the scent emitted from a decomposing body, are released. Blowflies are alerted to the potential, right? They have a very, very strong sense of smell, right? So they can smell that for miles, okay? So for miles around, and then they come in and lay their eggs. And then after feasting on those protein-rich fluids from the decomposing body, then they, female flies deposit their eggs in clusters on their body. All right, so blowflies are known as carrion, right? Carrion usually refers to what? So then that is going to be basically a body is carrion, right? So if you see a squirrel, like a squirrel roadkill would be carrion. We have our bottle flies, and we have blue bottle, green bottle flies, right? So these are the two distinctive kind of flies, and you've probably seen similar looking flies flying around. All right, so during the life cycle of the blowfly, right? So we here have our life cycle of the blowfly, right? So we have egg mass first, and then we have first, second, and third instar, and then pupa and adult, right? So we have first, second, and third instar, and then we have our pupa, and then now we have our adult, right? So this is the life cycle of the blowfly, right? And so then that just kind of keeps going through, right? That's the complete metamorphosis, right? Just like a caterpillar becomes a butterfly, right? That's another kind of metamorphosis, right? So this is the blowfly life cycle metamorphosis. All right, so these are the times, right? This is how we can estimate the time of death, right? If we use the life cycle of the blowfly, right? Because the egg stage is really only 8 hours long. So if you're seeing just egg stage blowflies, you can say that that body is only there, has been dead within around 8 hours, right? So then you've got your larva stage, first appears after 1.8 days, right? Lasts 20 hours. So then we have our instar 2, right? So these are the 3 different larva phases, okay? Instar, right? Instar 1, 2, and 3. And we'll get more into that in a little bit. So you can use the first appearance and the duration of the phase and those characteristics, and you can get yourself a time of death. Not an exact time of death. It's going to be more of like an estimated time of death, right? Which is one way to establish time of death, which is important in solving a crime so that the police officers can then backtrack and see where had the victim been, who did they come in contact with, right? So then you can get that good old-fashioned police work in there. All right, so the blowfly larvae use hooks to kind of dig in and scrape. So when you see all of that movement, right, so their bodies move, but they also have these little hooks that hook into the flesh. And remember, maggots, these larvae really are only feasting on dead, decomposing flesh, right? So they do not harm living tissue, and they do not carry, they're not transmitting, disease. All right, so the body face, this spherical configuration alerts investigators to what development stage of the instar, right? So the spherical contains the slits for breathing, right? So that's one of your vocabulary words, right? So the spiracles are responsible for helping the insect breathe, and we have instar 1, 2, and 3, which has to do with if you look at it, 1 spherical, 2 spherical, 3 spherical. Okay. So here we have the empty pupal case, right? So in here we have a head emerging from the pupal case, right? So this is the pupa stage. The next stage after the pupa phase is the adult phase, right? So here we have an adult fly coming out of the pupal sac to complete the life cycle. So here we have the common housefly, right? So we can identify that by its dark stripes. We have a flesh fly and we have a coffin fly. So right, not all flies are equal, right? So there's much, variety among the fly kingdom, and an entomologist is going to use those to help identify location and time of death. So now there's other insects that come in and help with this decomposition of a human body or animal body back down to its parts. We also have beetles. Okay, so a beetle is not the same thing as a fly. And they also follow a very predictable path of insect succession, right? So then on a decomposing body, right? So here we have our beetle life cycle. We have the first instar, second instar, pupa, adult, okay? And then we have, here's some 4 families of beetles, right? So I know definitely I've seen these things before lying around. Oops, gotta go backwards, went too far. Okay, so here we go. All right, so here we have different beetles and approximately what time they would arrive, on a corpse, right? So very early in the stage, right, here we have the clown beetle, right? The clown beetle's preferred food is fly eggs, maggots. All right, so then, right, so now not only do the maggots decompose the flesh, right, so now we have beetles that eat on the maggots, right? This is all part of a cyclical food chain. And then if you find these beetles, right, the hide beetles, right, then you have a very advanced corpse on the, very dried remains, right? So that means that the time of death was going to be quite a long time ago. All right, so then what do these things all have to do? Why do we study entomology, right? Because we want postmortem interval, right? So PMI. So that's where the importance of the blowfly and the cycle, the life cycle, the metamorphosis of the blowfly comes into play. So there's factors that are going to affect the development of the metamorphosis, right? The temperature and the environmental conditions. So this past spring, in the fall, when we looked at our decomposing squirrel in the courtyard, right? The warmer the temperature and the more rain, we're going to have a faster decomposition process, right? And if it was colder, we would have a slower decomposition process. So then we can use the accumulated degree hours, which is the number of hours that an adjusted average Celsius temperature that it takes an insect species to reach a particular stage in development, right? So a forensic investigator, entomologist, if they're on the scene of a crime, right, they're going to have to take the temperature and they're going to look at the meteorological conditions, right? So they're going to be looking at wind, rain, humidity, and temperature and other environmental factors. So we have our death scene observations, right? The entomologist is going to collect, right? Is going to collect insects around the body. There's going to be two collections, right? We're going to collect the live insects, and we're going to collect and preserve other insects at the crime scene. And then after the body is removed, right, we're going to take insects from the surrounding soil, right? Because all of those things are going to be able to give us, some really good observation. And again, they're going to have to take that meteorological data. So in summary, insects are the most numerous of all animals, and they provide valuable evidence in solving crimes. And they're also really a very, very protein, right? Full of protein. Forensic entomology is used to estimate that post-mortem interval, right, using the geographic location of the crime scene. And then they can link to suspects, right? We can determine if a body was moved, injury sites, exposure to drugs or toxins, and provide evidence of neglect or abuse, right? And then we have our 5 stages of decomposition, right? The fresh stage, really nothing, no real noticeable changes when you first appear upon the corpse. Then we have our bloated stage, right? So where the decomposition process is started internally, right? And the bacteria start digesting some soft tissue and some organs, and then they're going to release some gases. Then we have our active decay stage, right? This is where definitely we have a lot of, big maggot activity in the active decay and the advanced decay. And then we have that dry decay series where you're basically just going to have some of those beetles. And we have predictable changes in the chemical and physical environment of a body in the area surrounding it. Forensic insects include, right, flies, beetles, wasps, ants, and more, right? So we mostly at this level are really looking at the flies. And they follow a very predictable sequence, right? So you're going to need to know that life cycle of insect succession. And the blowflies, right? So those are the ones that we're really focusing on, are usually the first to inhabit a dead body, right? By the scent of that cadaverine and the putrescine. And there's 4 stages to their metamorphosis, right? That life cycle that we've been talking about, right? So we have the first stage is the egg, right? So the adult fly lays the egg. Then we have our larva stage, right? The larva stage is separated into the instar stages, which has to do with the spiracles, right? First, second, and third, 1, 2, and 3 spiracles. And the larvae use mouth hooks to help they ingest food, and then they move, and then they store food and breathe. All right, beetles usually arrive after the flies, okay? Variables affecting the rate of insect growth, right? Temperature, sunshade, wind, moisture, injuries, whether it's clothed or wrapped, right? This is a couple of things that they study at the body farm, if you've heard of the body farm before, which will be one of the activities we're going to look at in this unit. The post-mortem interval, right, is the estimate of the time between when the body was found and death. Accumulated degree hours, right, the number of hours at an adjusted average temperature that it takes for an insect species to develop to a given developmental stage. This accumulated degree hours is used to estimate the post-mortem interval. And then our evidence collection at the crime scene, right, they're going to collect live insects and their eggs. They're going to preserve those insects, usually in alcohol, and they're going to also collect that meteorological data because that's going to help them establish the most important thing that they're really looking for here, post-mortem interval and time of death. All right, that is it. Until our next time.